Hello everyone, this is Tanya Joshi of MSc Microbiology second semester presenting on the topic DNA and its historical experiments. These are the contents. Now let us first talk about what are nucleic acids. In 1889, Richard Altman obtained the first protein-free material to which he gave the name nucleic acid. These nucleic acids are the polymer of nucleotides, therefore are also called polynucleotides. They are the very large molecules which ranges from 21 nucleotides to up to a large chromosomes. There are two types of nucleic acids that are DNA, deoxyribonucleic acid, which is a double-stranded molecule having cytosine, guanine, adenine and thymine bases. Second is the RNA, which is ribonucleic acid, is single-stranded molecule having cytosine, guanine, adenine and uracil bases. Now let us talk about nucleotides. Nucleotides are the monomeric units of nucleic acids. They are phosphate esters of nucleoside and made up of three components that are nitrogen bases, that is a base which has a nitrogen atom, and these are purines and pyrimidines. Second is pentose sugar, which is a 5-carbon sugar, and nucleic acids like RNA and DNA have ribose sugar and deoxyribose sugar respectively. Third is the phosphate group, which is an ion of phosphoric acid, and molecules without phosphate group are called as nucleoside. Now the nitrogen bases are the hydrocyclic, planar and relatively water-insoluble aromatic molecules. These are two types in both DNA and RNA as purines and pyrimidines, wherein purines are two-ringed heterocyclic nitrogenous compound having 4 nitrogen at 1', 3', 7' and 9' position respectively. These includes guanine, that is 6-oxy-2-aminopurine, and adenine, that is 6-aminopurine, while pyrimidines are single-ring nitrogen compounds having 2 nitrogen atoms at 1 and 3' position. These include cytosine, that is 2-oxy-4-aminopyrimidine, thymine, which is found in DNA, is 5-methyl-2,4-dioxypyrimidine, and uracil, which is found in RNA, is 2,4-dioxypyrimidine. Now DNA, which stands for deoxyribonucleic acid, majorly exists in double-stranded form. It is located inside the cell within a nucleus. DNA acts as genetic material and stores the genetic information of life form. It encodes the sequence of amino acid residues in proteins using the genetic code. DNA is arranged or packed in a structure called chromosome. Basically, DNA is a large linear polymer composed of 4 kinds of monomeric units called deoxyribonucleotides having adenine, guanine, thymine and cytosine linked to each other in a chain. Now let us see the structure of DNA. The double helical structure of DNA was first described by Watson and Crick in 1953 and were awarded Nobel Prize for it in 1962. The Watson-Crick double helix model describes the feature of B form of DNA, which includes that 2 long polynucleotide strands are coiled around a central axis. Strands are wrapped in a right-handed helix conformation. Strands are antipolar, that is, one strand is 5' to 3' oriented and another in 3' to 5' orientation. Strands interact by hydrogen bond between the complementary bases, wherein the guanine form 3 hydrogen bond with cytosine and thymine form 2 hydrogen bond with adenine. Helix diameter is found to be 2 nanometer, or we can say 20 angstrom. Angle of interaction between base pairs result in major and minor grooves. Axial height of one helical turn is found to be 3.4 nanometer and 10.4 bases pair are present in per helical turn, and the distance between two adjacent bases is 0.34 nanometer. Base pairs are inside of molecule stacked close to each other. Now let us see the X-ray diffraction image of DNA. Rosalind Franklin, along with Maurice Wilkins, experimented X-ray diffraction to understand the physical structure of DNA and found it to be in B form. Here the figure represents the X-ray diffraction pattern, wherein the spots forming cross in the center denotes a helical structure and the heavy bends at the right and left arises from a recurring base. From the X-ray diffraction, Dr. Franklin deduced the actual structure of helical of DNA and its diameter, which is found to be 20 angstrom. She also suggested that the sugar phosphate groups were outside and the backbone of A form is antipolar. Here the diffracted rays interface each other, which results in overlapping circles. Now let us discuss the type of DNA based on the location, which could be nuclear DNA or mitochondrial DNA or the DNA which is present inside of plastids of plant cells. Now let us see the difference between these two. Nuclear DNA is located within the nucleus of eukaryotic cell while the mitochondrial DNA is inside of mitochondria. Nuclear DNA usually has 2 copies per cell while the mitochondrial DNA has 100 to 1000 copies per cell. The structure of nuclear DNA chromosomes is linear while the structure of mitochondrial one is closed and circular. Nuclear DNA is diploid while the mitochondrial DNA is haploid. Mutation rate is less than 0.3% in nuclear DNA while the mutation rate is generally higher than that of nuclear DNA. Now let us see the different forms of DNA, which could be A, B and Z form. First is the B form. It is the most common form of DNA. It is right-handed helix, turns after every 3.4 nanometer. Here each turn contains 10 bases and distance between each two successive bases found to be 0.34 nanometer. B form of DNA contains two grooves, major groove which is wide and provide easy access to bases. Another is minor groove which is a narrow one. Now the A form of DNA. It is a less common form of DNA but more common in RNA. It is also a right-handed helix. Here each turn contains 11 base pair per turn. It is also having two grooves, major groove which is very deep and narrow, and second is the minor groove which is very shallow and wide. Now the Z form of DNA, which is a radical change form of B. It is the left-handed helix. It has GC region-rich DNA. Here sugar base backbone forms zigzag shape. Now let us see the functions of DNA. Major functions include the replication process, storing genetic information, directing protein synthesis, determining genetic code, mutation or in the process of recombination, in gene expression, responsible for the metabolic activities like evolution, heredity and differentiation because DNA is a stable molecule and holds most complex information for longer period of time.